Dr. Arumugam from CSIR West, Thiruvananthapuram, and he's going to talk on selenoproteins from microalgae, basic biology and application.
Thanks for the chair for the kind introduction. So I am from CSIR NIST Thiruvananthapuram, and I will be talking today about the selenoproteins, which is a unique biomolecule from microalga, its basic biology as well as the applications. As I told, this is my institute, and for the student we have a SSAR academic program for the PhD admissions. So those who are qualified the national exam like JRF from different CSIR, DBT, those are all eligible to apply, and we do have 5 disciplines. Chemistry, this is a chemistry cluster tab, chemistry being a primary research. There are other activities as well, those are including biology. So I will be talking 3 things. First of all, the basic biology about selenoproteins. As I told, it's a unique because its synthesis has some special features. So all those we will be discussing. And then the selenoprotein as such, we found an organic selenium source, that is in the form of selenocysteine or selenomethionine or entire selenoprotein, which are non-enzymatic. So that process is a patented process. And then the toxicity, bioavailability, and the bit of therapeutic applications. It is needless to introduce the microalgae to the learned people, but for the sake of student, it is a photosynthetic organism. Microalgae is a photosynthetic organism which fixes the atmospheric carbon dioxide using the sunlight through the energy efficient process called photosynthesis, and it is having array of products, lipids to many pigmented proteins, all those things. And I will be working on omega-3 fatty acid as well, but that is not the topic of today's talk, but more of selenoprotein. And this microalgae will save the future because the farming land and other things are shrinking. So this will give a helping hand for us to secure a sustainable production of food product as well as the other therapeutic compounds. So this is the selenoproteins. It is selenoprotein. It's called selenoprotein because the selenium is co-translationally incorporated into the protein, which is essential trace element. It's quite often it was believed that it's a toxic chemical. Quite often there was an incident in the southern province of China wherein Keskan disease, the bone deformities developed. People tried out, even the physician tried to figure out what it is, but they couldn't. But later they supplemented the selenium, they are completely cured. The reason being that those regional people who have consumed the food, the plants which is grown in that soil, which lack of the selenium. That is why, because the selenoprotein machinery is lack in the plants. However, it accumulates, bioaccumulation is happening. So those regions which is lacking the selenium, the people suffered with this disease. From then onwards, it gained importance, biological importance, that it is having physiological relevance with many diseases. And then these functions is performed by the inside the organism by the class of protein called selenoproteins. The selenoproteins, the classical Watson-Crick genetic code, we used to see 61 amino acids plus 3 stop codon, but it won't behave that one because this selenocysteine is incorporated later. I will come to that bit later. Okay, the key function of this enzyme is it maintains the redox balance and signaling as a primary antioxidant system. Because the one reaction which keep on happening is reduction. So that is why it's very important to maintain the redox balance. These enzymes are very essential. Although it have other mood-like functions, but this is one of the key function of these proteins. In addition to that, it have a variety of other functions by family of selenoproteins. Many functions are there: thyroid metabolism, reproductive health, immune system, and anti-defense system is one of the prime function of these proteins. Yeah, something missing, that no issue. There is a selenoprotein family, many proteins are there. I am working on selenoprotein T, which belongs to the family of thyroxine like superfamily of proteins. And this is the classic motif, cytosine, followed by U stands for the seranocysteine, which is incorporated co-translation. Yes, I told you this PESEL. You can see that the TGA is otherwise called stop codon. The student, they will be familiar for them, it will be more useful as well. The TGA in frame, ATG start codon is there, followed by TGA. Okay. So otherwise the protein translation machinery will be stalled at the time, because it senses as there is a stop signal. I must stop here, but here it is not stopping. How it is done? Because at the 3' UTR, you can see here that 3' UTR we mapped all those things. So that is the story all about. So there is a PESEL structure. This is called a SEC machinery or seranocysteine insertion sequences. SEC-IS, which stands for seranocysteine insertion sequences. It is a machinery. These structures finds the RNA polymerase not to stop there. Also, afterwards, it will be recruited a group of proteins which all put together. That instead of stopping there, it brings there is a specific tRNA for seranocysteine. It brings the seranocysteine and incorporate there. That is how the machinery continues. So we did initially, we were finding difficult by using some other homology models. We designed a variety of primers up and upstream and downstream, everything, nothing worked. Then finally we went, because this microalgae genetic information is unknown. So the whole genome we sequenced and found that all the sequences, that is where we got it. And later on, the right primer could amplify the specific regions. So in addition to the specific the SEC insertion sequences, there are other elements like tRNA for seranocysteine, that is specific to seranocysteine T, and then other tRNA synthase and phosphoryltRNA kinase. These are all the machinery associated. All the things in microalga experimentally we have found out for the first time, and that is an advantage, and which has individually every functions. Since it's known in bacterial system, only with the sequence level we have found out that this machinery lacks in the plant system. That is why it cannot synthesize the seranoprotein of its own, but the microalgae can do that. Yeah, then we mapped it. It has the 3 introns and 3 exons, all the things, and at the 3' UTR you can see the secrets element, which is 94 base pair length, and then this is what once again I am emphasizing that this is the secrets element for the selenoprotein T, and there is an intergenic region of around 163 base pair followed by secrets element is there, which forms the classical hairpin loop like structure, which senses the RNA polymerase not to stop at the stop codon. Instead, it brings the seranocysteine to incorporate for translation. So along with there is a two organism. One is a semidecimus quadriquadal, one is Nanochlorhexis oceanica. So there is a minor genetic information is known for Nanochlorhexis oceanica. So we used that as a reference strain to identify in our own strain. And then the other model like lambda models also, we have extended this one. We found that it is fairly conserved among these organisms. So whatever that we developed, the model is varied, and this which we published in a relevant journal. And this is the secrets element regions for the seranoprotein T. We mapped it and compared with the other eukaryotic or the non-eukaryotic instead of seranoprotein T as well as seranoprotein W as well. We have that. This is the specific tRNA with the anticodon and acceptor loop, all those things. And this is very well conserved between semidecimus quadriquadal and oceanica. And this is diversity of this one. Yeah. So the functional aspect is that in addition to its classical function of maintaining the redox balance, it also found in ER stress. There is from some fungus. People have shown that it is having rolling ER stress. So we found that we just mimicked that, we induced the ER stress by treating with the DTT, and found that the expression is almost 2.2-fold more expressions were found through quantitative RT-PCR. So it does has, but it has an opportunity. We are exploring in that direction further. And then when we come to the second aspect, I am working for CSAR. CSAR, the basic biology is fine for academic interest. but we need to have some kind of product, industry-oriented research. So they want so for that one we developed this as an organic selenium source. And we carefully evaluated that using ICP-MS. And here you can see the development. It was not a small task. Over several generations, it's being this strain being developed. You can see that the first this is the control, and with less lag, it stops and goes to the normal growth phase of exponential growth, followed by the dead phase. And similarly, at lower concentration also, when we increase the concentration of the sodium selenite, there is a prolonged lag period. And people even found out that the student lost interest or something, that of late it's again started growing, because that is a physiologically unusual concentration. This organism found that something toxic to it, but however it maintained it. We know from the other yeast and other strains that it is being selenoprotein being done. So it took prolonged lag time, and now we developed over several generations. It's a sequential things we developed for over several generations, and then later we found that now we have a strain which can withstand up to 1000 micromolar. These are a bit old data, but up to 1000 micromolar of organic selenium can withstand without any lag. It can grow from like a normal strain. Otherwise, even 5 millimolar is very unusual for the other organisms. That is an advantage. Then later on, we found that it's growing fine. That does it accumulate the selenium inside or not? We evaluated through ICP-MS. We found that almost 1200 ppm of organic selenium. This is with the 500 micromolar, so we have some improvement over that. In addition to that, it also has the essential omega-3 fatty acid EPA, eicosapentaenoic acid, which is having its own clinical relevance as a cholesterol reduction and all those things. And this particular process, we filed a patent. It's a filed one in India, US, as well as the PCT for European Union. Here again, I am not going further, but we found that this strain is withstanding up to that much sodium selenite. What could be its genetic architecture, whether any changes happened, what made them to withstand that? All those we have made a comparative transcriptome, and those data I am not discussing here. So I move to the scale up. As Professor mentioned, that whatever we are seeing in the flask is quite different from the actual scenario. So we need to scale it up. We have the 50-liter reactor, photobioreactor. So for all our studies and further animal study and the company, this being validated to the company as well. So we are scaled up in each reactor. Maximum of 50 liter can be cultured. So up to 200 liter. It is when we are doing that in the flask, it could be with a little optimization. We can scale it as well. Those same 1000 micromolar concentration, the organism can grow in relatively large scale. I cannot call it as a bigger reactor, but when it works in 50 liter, I hope it will work in 1000 liter as well. Yeah, okay. So the toxicity and bioavailability, we have to test it out, and we do not have in-house animal house facility. So before going for that, the microalgae having one intrinsic problem is that it has the recalcitrant cell wall. Whatever unlike bacteria secrete out, it doesn't secrete out. It stays inside. So this it's a 3-layer cell wall made up of cellulase and pectinase, and in between some other layers which is poorly characterized. So we made some effort to carefully optimize the concentration of these enzymes, pectinase and cellulase, and we found that completely without leaching out, we can logically with the concentration of the enzymes we can manage to remove or break the cell wall. Thereby, it is the ruminant animals or other one consumes it is whatever the bioconstituent will be available to the organisms. So after doing this one, and then we went ahead with the animal study to evaluate this organic selenium. Does it have any toxicity? Because selenium is it's like a double sword. It's toxic as well, beyond certain concentration. So we went ahead. One is a normal study, toxicity, bioavailability, growth gain, any mortality, all those things. The other one is challenge. We fed this one for about 8 weeks, and then we challenged with we introduced the classical for this protein antioxidant effect is there. So we given the chromium induced anti-oxidative stress we have given and fed that one. It performs very well there. So you can see the data. It doesn't have any toxicity. as well as it's more bioavailable because, as evident from the serum selenium level and other different organs. Everything we analyzed it through ICP-MS, and we found that it is accumulated serum in a physiologically relevant level. And then the basic lipid profile, and the outcome of these studies are that it reduces the cholesterol. It doesn't have any effect on EAG and high density and other very low density lipids, whereas it reduces the low density lipid, which is not good for the health. But other effects we do not found any effect on that. Similarly, the hematology as well. The thing is that it elevates the serum selenium level. I told you through ICP-MS we found that one, and it elevates the selenium level in other tissues as well, and it is very significant increase in WBC was observed, and this organic selenium reduces the oxidative stress. Here I have not shown the data. There are enzymatic and non-enzymatic like glutathione peroxidase, catalases, and those things all we have evaluated, but since the time is restricted I am not showing that. And the lipid peroxidation is the hallmark of ROS reactive oxygen species. Right? Almost when the lipid stress is the oxidative stress started, there is a scavengers also activating like catalases, MDA level, all those things we have tested out. It seems to be that the organic selenium enriched microalgal biomass having a good potential to act as a human supplement as well as the food feed ingredient supplements. So the conclusion and future perspective is that we decoded the selenoprotein P and it associates set insertion machinery. And if anybody interested in selenoprotein P, T, the family of proteins are there. If you want to express that one, in normal vector we cannot express it because these machineries are lacking in other things. So these microalgae can be used as a heterologous expression system to express other human selenoprotein P and other things. And then the structural validation being done, and we enriched the organic selenium, enriched the microalgal biomass, and then the recalcitrant cell wall also being selectively. It's a kind of package. Organic selenium is there, and the cell wall can be removed. So it is ready for dietary food feed supplements. If any industry people list I found here, if at all in future collaboration as a perspective that industry people are there, we can have a valid MOU and we can discuss and see its feasibility for their applications, and also any for therapeutic applications appropriate cell line model, and we do not have enough cell line model. So somebody, the like-minded people are there, we can collaborate to go further in this aspect. So with this, I thank my student Reshma. It is she is a very sincere student and intelligent student. All the work which he done, she is also one of the co-inventor of this patent. And Reshma and all my funding and team IMC for inviting me to share some of my talk. And thank you all for patience. I'm happy to take
Anybody has any question? One question, if you have.
Can I ask one question? Did you look at the liver function test in the mouse models?
No ma'am, not yet. I am planning to that liver NASH or some fibrosis model is there. So those models someone is having, we can do it.
Selenium related.
Yes, exactly. And also we can test it out. That is one of my interests. And also if the endocrine related function, some specific things, I made some effort because of non-availability of suitable partner there. It's ours is a biotechnology division is a little, it's a chemistry cluster lab in CSIR. We do not have that much biological activity, widespread activity, and animal model is one of the things which lacks. In vitro assays, I don't believe all those things, so it doesn't work beyond publications. So if we have a suitable model, we can collaborate.
Thank you so much.
And this biomass is now we can scale it up in a gram scale. How much ever amount we want throughout the field trial we can conduct and arrive at meaningful conclusions.
All the best.
Thank you.
Thank you. So, if there are no questions,
Thank you, Dr.
thank you so much.
Thank you, sir. Thanks for the wonderful presentation. Now I request Professor Kalpana Pai to present a moment to Dr. Anju.
Yeah, please. Thank you.
Thank you.